All right, today we are going to look at topic 1.6, DNA and RNA. And while they both carry genetic information, there are quite a few differences between these two molecules. So I'm going to do a comparison of the differences between DNA and RNA and then a quick summary at the end, all right? So I'd like you to fill out your tables as you listen to this video. And you can find this video linked on PowerSchool under today's review PowerPoint. And then you can either just make this table in your own notes or wherever you take your notes, or you can do it just digitally here. I'm going to leave that up to you because this is your notes to use. All right, so the first difference is the names of DNA and RNA. So the name of DNA is deoxyribonucleic acid. All right, two separate words, deoxyribonucleic and then acid. RNA is just ribonucleic acid. All right, and we're going to talk about why these two differences in names. But this is our first difference. Second difference is the function. All right, so DNA, the function of DNA is long-term storage of genetic information. All right, and it uses this information then to make cells or make other cells and new organisms. DNA doesn't necessarily do this, but it's utilizing this genetic information to make new cells and new organisms. All right, then looking at RNA, different structure, so different function. It's used to transfer the genetic code from the nucleus to ribosomes. Now I know we have not covered structure of cells yet, but you will figure this out next unit, all right? RNA, and then ribosomes to make proteins. So it transfers this genetic code provided by DNA. And RNA, as we've talked about in the RNA world hypothesis, it may also transmit info in some organisms, and may have been used to store genetic blueprints. A blueprint, a blueprint is the information off of which we build a house or a structure. It could have been used to store genetic blueprints in primitive organisms. All right, so our second difference is function. Okay, second one is talking about the structural features of DNA and RNA. Note that they're backwards on this, so take that into account. DNA and then RNA. All right, so DNA forms what we call a B-form double helix. This is just the form of the helix. You don't necessarily need to know that the interaction of the minor and major groups, but just know that it's a B-form helix. Whereas RNA is going to form what we call an A-form helix, and that's just simply how the phosphate backbone is organized such that it forms a certain type of helix. So we have DNA is B-form, RNA is A-form helix. Okay. DNA is also double-stranded. As you can see here, we have two strands of DNA, whereas RNA is single-stranded. All right. And it's going to RNA also is going to be shorter. Contains shorter chains of nucleotides, whereas DNA is going to contain long chains of nucleotides. And there's different structural differences here, or more structural differences here, in the composition of bases and sugars. So for DNA and RNA, DNA is going to be composed of a deoxyribose sugar, while RNA only has a ribose sugar. All right, the difference here is there's one extra hydroxyl group on the RNA that is not present in the DNA. That makes all of the difference. All right, next difference, DNA has a phosphate backbone. RNA has a phosphate backbone. But the third piece here, okay, so this is a difference, or a similarity. The big difference here are the nucleobases. The nucleobases for DNA are adenine, thymine, guanine, and cytosine. All right, and actually RNA is composed of 4 nucleobases. And you can also call these nitrogenous bases, sorry. So you can say nucleobases or nitrogenous bases, or simply bases. But in a test situation, they're going to probably say nitrogenous base or nucleobase. Our nucleobase for RNA is also adenine. I'm going to skip guanine, cytosine, cytosine. And the difference here, it does not have thymine, it has uracil. All right, that is a different base that only RNA has. All right, and here's that picture again, but just a reminder, backwards, I don't know how to switch it, I'm sorry. But here we have for RNA, you can see our cytosine, guanine, adenine, and uracil. For DNA, we've got our cytosine, guanine, thymine. Sorry, this looks like it's backwards. Oh, adenine and thymine. All right, so nucleases here are different. However, know that uracil is still a pyrimidine. Just because it is different, has a little bit different structure than thymine, it still is going to be a pyrimidine, which means that it will only form 2 bonds with adenine, and this is a weaker bond than one between cytosine and guanine, which has 3 bonds, adenine and thymine, 2 bonds. Okay, so pyrimidines and these are called purines. And this bond is going to be much stronger than this bond because there's 3 bonds between those nucleobases, whereas this one only has 2 bonds. Next difference is propagation. What does propagation mean? And usually when we talk about propagation, we're talking about plants, when you're growing a new plant from like seeds, from cuttings, or from other plant parts. But we can also talk about the propagation of DNA. How do we get new DNA and RNA? DNA, it's self-replicating. Okay, so how do we get new DNA? From the old DNA. However, RNA is different. RNA is different. It is synthesized from DNA on an as needed basis. So you have all the parts and pieces of RNA are just kind of floating around in the cell. And if we need to make a protein, or if we need to if DNA needs to send a message of its structure to another part of the cell, it will build RNA as needed from the code provided by DNA. All right, so DNA is always present in the cell. It's unwound in the nucleus, usually. It's not yet wound up into chromosomes, but it's always pre-built in the cell, whereas RNA is not yet synthesized and it's only formed on an as-needed basis. Next difference, base pairing. All right, kind of mentioned this earlier in DNA versus RNA. DNA has adenine and thymine are going to pair, and guanine and cytosine are going to pair. In RNA, adenine and uracil are going to pair up, and guanine and cytosine are going to pair up. All right, so the only difference here is thymine and uracil. Okay. Next, we're going to look at the reactivity of these two nucleic acids. The carbon-hydrogen bonds in DNA make it fairly stable. Okay, DNA is fairly stable. Plus, the body is going to destroy enzymes that would attack DNA. All right, so the body provides protection to some degree. It's not perfect. And also DNA, because it's the B helix, it has smaller grooves. So less of those bases are exposed, or fewer of the bases are exposed, and so this serves as protection, which provides minimal surface area for enzymes that would attack this DNA. There's less area for them to attach. Now when we're looking at RNA, that's an R, I promise. RNA, the OH bond, okay, remember we have that oxyribose, or the RNA has this hydroxyl group, so this makes it more reactive. This hydroxyl group is going to want to react. So it's more reactive and less stable than DNA. It is not stable under alkaline conditions. What is an alkaline condition? And this is another way of saying high pH. All right, so if you have a pH of typically greater than 10, this is going to destabilize our base pairing. So uracil. So at this point, if we have a higher pH of 10, this is going to destabilize our bases, and the RNA is going to break apart. All right. Now you might be wondering, Mrs. Schussler, I thought the RNA was single-stranded, and while RNA is a single-stranded molecule and can exist as a single strand, sometimes it will form complementary base pairing, which is when we'll see this base pairing, and which is when it would be denatured. In, for example, tRNA. Okay, an example of tRNA is this is going to be a double-stranded RNA molecule, but it is not always double-stranded. So that's just one caveat I want to make. Thinking back, RNA, less stable, and it's constantly being, here, I'm going to put this down here. It's constantly being produced, used, recycled, and degraded. Okay, so RNA is less stable. It's being recycled, produced, used, degraded, and one reason for that is because it has this A helix, this alpha helix. It has grooves in the molecule that make it more susceptible to enzymes attaching onto it and destroying those nucleic bases, right, and breaking those bonds. So the larger grooves provide less protection. All that to say, RNA is less stable, DNA is more stable or less reactive. And then an example, just to kind of show you the difference in the surface area, you can see here, the distance between here and here is much less on DNA, much greater on RNA, providing much more room for an enzyme to attach, whereas an enzyme here is going to have a harder time getting in because this phosphate backbone is protecting it. Last point, this one's pretty short, UV damage, because you might have heard that if you're exposed to UV rays, that this is going to increase your chances of getting cancer, and this is because DNA is susceptible to UV damage. which is ultraviolet light. Compared with RNA, or compared to DNA, RNA is fairly resistant to UV damage. Does it get damaged at times? Yes, but it is much less, or is much more resistant than DNA. That is our last difference that we're going to look at. In summary, I'm just going to summarize for you guys. DNA contains deoxyribose. RNA contains ribose. Ribose is different because it has one more hydroxyl group than deoxyribose. DNA is double-stranded. RNA is single-stranded. DNA is stable under alkaline conditions, which is our high pH. RNA is unstable during alkaline conditions. DNA, the function is the storage and transfer of genetic information, while RNA is going to code for amino acids and act as a messenger between DNA and the ribosomes for protein synthesis. Finally, thinking more about the structure, DNA is composed of nucleobases ATCG, while RNA is composed of nucleobases AUCG. Difference here, uracil lacks a methyl group on its ring, so if this were its ring. The only difference here is that it does not have this methyl group that T does have, that thymine does have, and uracil does not have it. So that's the only difference there, but that changes everything about that molecule. All right, so those are our differences. Go ahead and finish up recording the differences in your table. And then what I'm going to have you do next is I would like you to have a discussion with your partner. I would like you to pick a structural similarity. Don't pick the same one as your partner, but pick a structural similarity. And then pick a structural difference. So a structural similarity and a structural difference. I would like you to explain how those differences and similarities play a role in the function of DNA and RNA. Okay, so pick a similarity and a difference. How does this similarity play a role in DNA and RNA? And then how does this difference play a role in DNA and RNA? All right, so thinking about those two nucleic acids, what are the similarities and differences, and then explain the structural difference and the role it plays. All right, you can record this under your in your notes under your table. All right, thank you for listening. Bye-bye.